Hello class, I just wanted to show you how to set up today's notes. So I'm working from pages 84 and 85 in the science notebook. Please go ahead and update your table of contents. You can pause the video if you need to. So it's going to be Meiosis Notes on page 84 and Meiosis Output on page 85. Also at the top of your paper, on the left side, page 84, please go ahead and update your key in the corner, which is just kind of a list of vocabulary words that you're going to need to know in order to take these notes. Many of them you already know. In fact, all of them you should already know, but it's just a good reminder. And then over here is going to be sort of a title with also a little mnemonic for how to remember the difference between mitosis and meiosis. Specifically that meiosis makes me, and you'll see M-E in the front of the word. It's just a good way to remember it. This has to do with sexual reproduction and not asexual, so it's a helpful tip. So we're going to talk first about meiosis 1, because we're going to go in chronological order of what happens. The first thing that happens is interphase 1. That word should sound pretty familiar to you from mitosis. Basically, the contents of the cell double. That should sound familiar. So instead of being 46 chromatid, it becomes 46 chromosomes, or sister chromatid. So I'm actually going to use a different color. So I advise you use a different color too. I'm going to select red. So each chromatid doubles and becomes a chromosome. I'm going to use blue as well. And bam, you have all kinds of different sets of chromosomes. 46 of them in total. So let's go ahead and say 46 total chromosomes. Okay. Now, there's also our lovely centrioles in there as well. So during prophase 1, we still have the nucleus. The centrioles have moved to opposite sides, and our lovely, chromosomes, they actually find a buddy, and they attach to the buddy. And it is whatever chromosomes inside that nucleus code for the same genes. As you know, you get a set of genes from one parent and a set of genes from the other to make you, and so this kind of was the same way. So they kind of find a buddy, and that's all 46 chromosomes, so there are going to be 23 pairs of chromosomes in this case. Oh, and I forgot to include our spindle fibers. Can't forget about them. They start to make an appearance as well during prophase. Next thing that happens is metaphase, you called it, and we're going to give it the Roman numeral 1 afterwards as well, because it's metaphase 1. So again, cell, sorry my circles aren't perfect, centrioles. Now, here's where things get interesting. Each of the homologous pairs, now, of course, there's like way more than this, but let's pretend for the sake of fun and ease that there's just 4 sets. They line up down the middle with their buddy. And the sister chromatids attach to the buddies. And I'm going to write this on the side. These are called homologous. Now, you know the prefix homo means same. I'm really glad I can't hear my classes right now because I know that one gets you, but homo means same. So these are pairing up with their same gene codes, so the ones that code for the same thing. And they line up down the middle. And metaphase, remember M for middle. It's a good way to remember it. Okay, my favorite thing happens during this phase. So if I were to zoom in on, let's say, this pair, what you would see happening is the red is actually wherever the blue touches the red, it's going to swap genes with that piece of chromosome. It actually does a trade-off. Oops, no. And the same thing would happen for this one. A little bit of the red robes there. They do a little swapperooski. Now that is called crossing. Ooh, I don't like that. Oh, crossing over. And that is incredibly important because that helps us create diversity. If our cells did not do that when it actually divides, there would be very little diversity. And we've talked about why diversity is actually really a good thing in nature and can be very helpful for a species. So we like this stuff. This is super awesome. Okay, now we're going to go to anaphase. So anaphase, anaphase 1. And I bet people know where this is going. Now you've got our lovely spindle fibers. They are attached to the chromosomes, but they have pulled the buddies apart, which actually makes me a little bit sad. Now, just because I like to be accurate, I'm also going to draw a little bit of blue on each of these. Oops. It would make sense if the blue was on the parts that they actually touched. Here we go. And a little bit of red on this corner to show that they did some gene swapping so that there's diversity. So those get pulled apart. And telophase. One. So we have the, oops, no. Yes, we have our cleavage furrow that's happening. We have our nuclear membrane starts to form because we want our nuclei again. Of course, now there's 2. And over here, we're going to have our chromosomes. Here, we're going to have our chromosomes. These are going to have, oh, don't know what just happened there. Sorry about that. Technology is fun. Okay. Nope. Oh, whatever. You get the point. Okay. And then over here we have our red ones. This is a struggle. Okay. So that's our fantastic telophase. Now the centrioles, they are all wound up. There's no more spindle fibrils out there. And then you guessed it, cytokinesis, which means movement of the cytoplasm, is running out of room. Oh no. We have our 2 daughter cells. And I would show the crossing over, but that's too much. Okay, so you get the point. That is meiosis 1. So let's keep it simple. We basically are going to start meiosis 2 with what the cells look like at the end of cytokinesis from meiosis 1. But I'm going to simplify it. I'm not going to go phase by phase this time. Obviously, it looks very similar to mitosis because the chromosomes line up down the middle, single file this time. And when they're pulled apart to individual chromatid. So we have, just to kind of give you a high-level summary, so originally we had one cell at the start over here, and it went to 2, and now those 2 are about to become 4. So one cell became 4. That's right. It split and split again. Just if I were to take a piece of paper, rip it in half, and then rip those two pieces of paper in half and make 4, that's how math works. Miss Dag would be proud. Okay, so we have 1, 2, 3, 4, each with a nuclei. Now the tricky part is that instead of chromosomes, they have chromatid. Because remember, a sperm and egg only have half of what is needed. Now I'm going to be a little more accurate this time and actually draw some of the genetic variety that is left over from crossing over. Obviously it's way more complicated than this, but you get the gist. So you have 4. Now these each have 23 chromatid each. And that is because 23 plus 23 equals 46 chromosomes, which is the magic number to make a zygote or a fertilized egg. So it all comes back to that, because meiosis makes me. We have a little bit of a simple meiosis output for you today. So I'd like you to go ahead and label at the top of your notebook page on page 85, meiosis output. Write the mnemonic, meiosis makes me, underline the me in both. Draw a circle, and then inside that I would like you to draw a picture of yourself. You can make it about your family and your pets, but please make sure you are also a part of the picture. Add detail and color. Later you will be expected to share that on Flipgrid. Can't wait to see them. Bye.